Before watching this video, subscribe to my channel. A culture media is a special medium that supports the different organisms to grow by providing different types of nutrients. There are many types of organisms that have their own specific requirement of nutrients to grow. Classification of culture medium on the basis of their consistency: solid, semi-solid, liquid. Solid culture media. Solid medium contains agar at a concentration of 1.5 to 2.0% or the solidifying agar. It has physical structure. The solid medium allows culture to grow in the form of colonies or streak. It is useful for isolating the bacteria or determining the colony characteristics of the isolate. There are more than 100 of solid culture medium are available with different modifications. In this video, I am going to discuss principle, uses, composition, and colony morphological characteristics of the 26 agar media. Remaining culture media are going to discuss on my next video. Ethylene methylene blue media. It exists in the visual distinction of Escherichia coli. It can easily differentiate the lactose fermenters and the non-lactose fermenters. Cysteine, lactose, and electrolyte deficient media. It is recommended for the diagnostic urinary bacteriology. McConkie agar. McConkie agar is a selective medium for the isolation of gram-negative organisms. Gram-positive bacteria are inhibited by bile salts and the crystal voidate. The lactose fermentation is detected by pH indicator phenol red, which turns pink in acidic pH. Nutrient agar. Nutrient agar is a simple medium that is used for the growth of all types of non-fastidious organisms. Blood agar. It is specifically designed for the growth of fastidious organisms that do not prefer to grow on the normal media, such as streptococci. They require special types of enrichment. Chocolate agar. It is lysed blood agar that is used for the isolation of fastidious organisms. MUELLER-HINTON agar MLT. It allows the growth of all types of microorganisms. MANNITOL salt agar. It is specifically designed for the isolation of organisms that prefer high salt environment for their growth. Buffered charcoal yeast extract agar. It is a selective medium used for the isolation of Legionella pneumophilia. Bismuth sulfide agar is a selective medium for the isolation of Monila typhi and other species. Brain heart infusion agar. It is used for the isolation of fastidious organisms such as streptococci, meningococci, and Neisseria. Seborrheic dextrose agar. ST is a selective medium for the isolation of dermatophytes, fungi, and yeast. The growth of bacteria are inhibited by the addition of tetracycline, chloramphenicol, and gentamicin. But the filamentous bacteria can grow, such as Nocardia. SIMMONS citrate agar. It is specifically designed to check the organism's ability to utilize citrate as a sole carbon source and ammonium ions as a sole nitrogen. Inhibitory mold agar. It is a selective and enriched medium used for the isolation of fungi such as Histoplasma capsulatum, dermatophytes, and other pathogenic fungi. Brilliant green agar. It is selective as well as differential medium used for the isolation of Salmonella species except Salmonella typhi. Starch agar. It is non-selective and non-differential media. It is called differential on the basis of starch fermentation. Malt extract agar. Malt extract agar is a selective medium used to isolate and enumerate the yeast and molds. Sheep blood agar for the isolation of fastidious organisms such as Streptococcus pyogenes. Mead infusion agar. It is the nutritive medium used for the cultivation of organisms for the vaccine and toxin production. Potassium telluride agar. It is used for the cultivation and differentiate the Enterococcus faecalis. Thiosulfate citrate bile salt sucrose agar. It is a selective medium used for the isolation and cultivation of Vibrio species. Cysteine telluride blood agar. It is a selective medium used for the isolation and cultivation of Corynebacterium diphtheria. Buffet agar. It is used for precipitation test and immunodiffusion technique. Yeast extract phosphate medium. It is a selective medium used for the primary isolation of pathogenic dimorphic fungi. Xylose lysine deoxycholate agar. It is a selective medium used for the isolation of Salmonella and Shigella species. Tryptic soy agar. It is a universal medium that supports the growth of wide range of gram-positive and gram-negative organisms.